Hello, I'm Mr. Baker, and this is Unit 6, 6.2, Cellular Growth. The main idea, cells will grow until they reach their size limit, and then they've got a choice. We're done, or we're going to divide. But before they can divide, we have to understand cell size limitations, and that involves the ratio of surface area to volume. What is surface area? Surface area is the area of a solid object as a measure of the total area of the surface that object occupies. It's the covering, like your skin is your surface area. What is volume? Volume is the quantity of three-dimensional space enclosed by a closed surface. For example, the space that any substance, be it solid, liquid, gas, or plasma, or shape, occupies or contains. Volume, that's everything on your insides. There is a surface area to volume ratio, and as you increase the size of something, the surface area to volume ratio gets smaller and smaller. If you are a tiny particle and you want to get to the center of the baseball, it's a short journey. If you're a tiny particle, same exact size, and you want to get to the center of the basketball, it's a very long journey. This makes getting nutrients and expelling waste far more difficult, and your takeaway is that cells cannot grow very large. And here is a great video that demonstrate that you'll watch some other time. Once a cell reaches its size limit, again, it's going to stop growing or it's going to divide. Most cells will choose to divide, and that is how a cell reproduces, and we call that the cell cycle. One complete cycle gives us one cell that becomes two cells. There are three stages to the cell cycle. Interphase, where the cell grows, makes copy of its DNA in something called DNA replication, and the cell spends most of its time in interphase. Mitosis is when the cell nucleus divides, and cytokinesis is when the cytoplasm divides, which creates a new cell. It's represented by a diagram as such, but we have a nice little GIF for you. Into G2, where you're getting ready to divide, and then M is mitosis, where you do divide, and now you have two cells. So G1 is part of interphase, cells getting ready to go. S phase is when you make a copy of your DNA. G2 is when you're getting ready to finally divide everything. Mitosis phase is when you divide. Let's talk about interphase. There are three stages. Gap 1, synthesis, or gap 2, and they're known as G1, S, and G2. Gap 1, the cell grows and carries out its normal functions. Some cells never leave this phase, and they're no longer called G1. They are said to be stuck in G0. Muscle and nerve cells come to mind. Synthesis is when you make a copy of its DNA, and that's called DNA replication. And then we go to gap 2, and the cell's preparing for mitosis. And cells have what's called a checkpoint, and you got to have everything in order to pass this checkpoint to continue with cell division, because if it doesn't, then you've got cells dividing you don't want them to, and that can be a very bad thing. Main idea here is that DNA replicates by making a strand that is complementary to each original strand. And this is known as semi-conservative replication. New DNA molecules of one strand of original versus one strand of old. We have to unwind our DNA, and to do that we need some enzymes. One is called DNA helicase, and it unwinds and unzips the double helix. Each strand serving as a template for its new strand that's going to match to it. Then we have the molecule that does everything, DNA polymerase, adds nucleotides to the 3' end of each strand. It's making the copies, and you'll have two identical copies of the original double strand. Here is a good little video that you will watch some other time. You need to be aware of something called the leading strand, which is one that is built continuously, and then one of the strands can't be built continuously. That's called the lagging strand. The lagging strand has these little pieces on it that are called Okazaki fragments, and you've got to hook those together. This should help. Here comes helicase unwinding the DNA. You're actually going to put down a primer, then DNA polymerase shows up. Your leading strand gets to keep going up at the top. Your lagging strand has to build in little tiny pieces. And those are the Okazaki fragments on the bottom of that slide. There's another video that shows it in more detail, but We are going to watch that some other time. Here's a diagram, but the GIF does much better. There's an Amoeba Sisters video. And that's a lot to get through. You're going to rely on the videos to understand DNA replication, because it's something hard that's difficult to conceptualize just from listening to words. So go back and watch those videos. You can find them on the topic page on MyCC. These are the things that you'll need to know for your quiz. Study hard, and we will see you next time.